Good afternoon. Thank you for the opportunity to present ADARx Pharmaceuticals. We are the next generation RNA company. What makes us next generation is our proprietary technology. We have a data science-based platform that allows us to design cell-targeted delivery system as well as produce high-potent siRNA. And our candidates are designed to have deep knockdown effect, with most of them reach near complete inhibition of target gene, and they are highly precise and have long durability without integration into the genome. Our technology has attracted many top-tier investors, and recently we entered a collaboration with AbbVie, with the partner paying $335 million upfront and multiple billions upon milestone. This is the overview of our current pipeline, and today I would like to focus on geographic atrophy and ADARx 038. The 038 is a GalNAc conjugated complement factor B specific siRNA administered subcut. The GalNAc conjugation works as a GPS system, direct the product enter the hepatocytes. Upon the entering of cytoplasm, the siRNA will combine with intracellular machineries and build a risk compound, which specifically binds to the CFB mRNA and cleave it one by one and over again. This mechanism gives the drug high specificity as well as the very long durability. CFB is a potential treatment target for GA backed by genetic study and clinical evidence. CFB is a critical player in the alternative pathway activation, not only at the C3 activation, but also the downstream, forming a amplification level, a loop. This loop is thought to be associated with many pathological damages. Unlike targeting the terminal C5, this mechanism will leave the classical and lectin pathway intact, providing a better safety profile. We also have data demonstrating that systemic depletion of the CFB will lower the ocular level of CFB concentration, while making the targeting of liver-produced CFB a non-invasive method for the both eye. And therefore, we hypothesize that systemic depletion of CFB will slow the GA lesion growth. Our in vivo data in primates shows a single injection of ADARx 038 can reach near complete suppression of CFB protein production. Accordingly, near 100% suppression of the alternative pathway while leaving the classical pathway intact. As we also looked at the systemic depletion of CFB in the effect of local concentration of CFB in the ocular tissue, we also saw a similar reduction in the local tissue, not only in the retina as well as in the choroidal complex. If we look at the Western blot data, it is locally also reached near 90% of the reduction of protein level, similar to the systemic data. Our clinical data, phase 1 data, also demonstrated very similar, consistent result. A single injection of 038 reached 100%, near 100% alternative pathway suppression with a long durability for over 3 months, while the classical pathway maintained intact. The data also shows a very favorable safety profile for our compound, and we are in process of initiating phase 2 studies in GA and in multiple other indications as well. To have a better understanding of the inhibition level of our product can reach, here we compared our phase 1 study to the previous Golden study sponsored by Ionis Pharmaceuticals. We can see in the Golden study, the suppression of the CFB protein production is about 68%, while the inhibition of alternative pathway only reached 36%, leaving the majority of the alternative pathway active. So a short summary of ADARx 038. It is a potentially transformational treatment for patients with GA. This is highly potent and durable with a potential dosing interval for at least 3 months. So we are planning to start our enrollment for phase 2b study in GA next month. Thank you very much for your attention. We also have our CSO on site and our research and development team on site as well. If you are interested in any of our research and development program, please reach out to us. Thank you.